Hello. I'm going to explain a little bit about how we collect our marker-assisted DNA collections in the greenhouse today. And first of all, I want to say that every one of these crosses was made with a specific marker in mind, so that when we collect them and send them over to the lab in Pullman, they'll be looking for a specific characteristics that they were bred for. So hopefully we'll get a high percentage of ones that are desirable, but it also helps us select out the ones that are not. Because of the equipment in Pullman, we had to adapt our planting system to fit their collection system. So they use these DNA plates, and they're filled with these little tubes that have silica in them. And this desiccates the leaves and preserves the DNA as soon as it dries. And this has really streamlined the collection. We don't have to use liquid nitrogen and all this messy stuff anymore. It really streamlines it. So we have designed a system that matches this plate, which is basically an 8x12 plate. We collect a section. I'll collect a row at a time. Labeling is super important, so everything is marked with whatever the cross is and what plate it is, so that we can come back later and know exactly where we got it. When it's finished, we'll actually have a plate map that tells us which ones to keep and which ones to cull. So it makes it really easy to keep track, because as you can imagine, with this many plants, keeping track is a big deal. It doesn't do you any good to collect and not know that you got the exact plant. So what I have to do is I've got a little system, and I keep my hand oriented exactly how it's going to go back in. I want a leaf sample that is about this size. The exact size is not as critical. Too big, it's going to mold. Too little, they won't have enough material. So I collect one little piece from each plant and set it in the little container that it's going to go in. And we have found that if we jam it down too tightly, it molds, but if it's caught up in the top, it doesn't drop down, so they can get it into solution to get the DNA out. So we take tweezers and just press it part way down. So I don't know, we stick it into the tube so that it's part way down, so it's not going to get caught in the lid, and it's not all crammed in a bunch in the bottom. And then we always make sure that this is oriented properly, so we know that this matches up with this row, and put it back in. Okay? After I do a whole plate, I'll put them upside down so that the silica drops down, and make sure that the leaves dry properly, because they have to be dried so they can crush up to be able to extract them. We always leave 2 empty spots in the exact same place for every plate, and this is used as a control to make sure that they don't have any other background noise coming out when they're extracting. It's just to make sure that they're truly getting just the DNA from the plant. And so that's what I do.